Gene project, and this project was started by CSIR. And the intention of this project is to understand the extent of genetic variation in Indians. And here CSIR drafted 1,000 youth from the pool of about 5,000 Indians and included representatives from every state and diverse ethnicity. Means, see what exactly this statement is trying to make you understand. Means across the India, we made a pool of 5,000 Indians, right? From every state, from all ethnic groups, we have, collected, and out of the 5,000. The 1,000 youths were finally selected for the genome donation. From the genome donation, clear? From the genome donation means from the sequencing of the genome. Clear? Fine. So 5,000 across the India was the complete pool, and from the 5,000, only 1,000 were finally selected for the sequencing of the genome. Fine, and the intention is what? Just to find out the extent of the genetic variation. Okay, and this project was involved by Hyderabad-based CCMB, Center of Cellular and Molecular Biology, that's IGBI. That is the CSIR Institute of Genomics and Integrated Biology. It was a joint venture of CCMB and IGIB. That is the CSIR Institute of Genomics and Integrated Biology. Clear? Then next to this, one more project was started by India, and this project is what? It's a Genome India project. That is the Genome India project, and it's a 238 crore Genome India project. Will involve 20 leading institutions, including IITs, IISc, Bangalore, and IIT, means Indian Institute of Science, Bangalore, and IITs. And in this. We are supposed to frame a grid of 10,000 persons from all over the country. So the initial project was include 1,000 youth across the India. But now in this project, we are trying to maintain the sample of the 10,000 persons from all over the country to form a grid for the genome. And that will consider as a reference genome, that we need to maintain the gene pool of 10,000 Indians. Right, and the data will be stored, and that data is considered as a reference genome. Clear? Fine. And why this need for the genome sequencing is required? Because the ultimate purpose is what? Mapping of the diversity of the Indian genetic pool will lay a bedrock of personalizing medicine and put it on the global. Means ultimate purpose is what? The ultimate purpose is this will be acting like a research tool for finding the the cause of genetic disorder or the metabolic disorder or the complex disorder, like the diabetes, like the heart, like the diabetes, like the mental problem, like any kind of a disease, right? And then after knowing the basic aspect of a particular disease, we should try to find out the medication. Clear? We should try to find out the medication. Means we will try to find out the right distinct medication for the curing of that particular disease. Like, see example, when we talk about the diabetes, for the diabetes we don't have a specific medication, right? Diabetes, we don't have permanent medication for the diabetes, and that is the standby medication by the use of the insulin. But insulin is not a permanent medication. Fine. So if we are able to sequence the insulin gene that already we have sequenced, right? Then if we are able to see how this mutation is going to be done in the insulin gene, and how this insulin gene is going to be inactive or hyperactive. So if we are going to justify everything, so we'll try to find out the fixed and the final way for the medication, permanent way for the medication, for the medication of the diabetes. So like this, the ultimate purpose of the Genome India project is what? Just to find out the cause and the medication of the complex disorders like diabetes, mental health, or even the genetic disorder. Okay, fine. Again, one more project was launched. And that is what? It's a National Genomic Grid. And this National Genomic Grid is what? It's a grid for the cancer patient. It's a reference genome for the cancer patient. Right? So this National Genomic Grid, this will collect sample from the cancer patient across the India. And here the cancer gene sample will be maintained in the National Cancer Tissue Biobank, which is set up at Indian Institute of Technology, Madras. Right, and the biobank collect cancer tissue sample with the consent from the patient diagnosed with the cancer. So the ultimate purpose of National Genomic Grid is just to maintain a grid of cancer patient so that they will go for the more research, exploration, and the better medication for the treatment of the cancer. So this National Genomic Grid is basically dedicated for the research in the cancer as well as finding a better medication for the cancer patient. Fine. So we could see here application is what? Here the aim is to provide researcher with the high quality of the cancer tissues and the patient data in order to facilitate cancer research that will lead to improvement in the cancer diagnosis and the treatment. So ultimate purpose is what? It's a research and medication for cancer patient. Research and medication for cancer patient is the ultimate purpose of National Genomic Grid. Is that clear? Fine. So what we have done so far for the human genomics, one is the Human Genome Project. Again, there was so many independent projects that was launched by independent countries that we have mentioned for the prelims. They are not going to ask, these statements or these points in mains. Again, indigenous project, especially in the Indian context. Fine. The Genome Indigenous project, that is the Indian project. Genome India project. Then National Genomic Grid. National Genomic Grid. And see, when we talk about the prospect of the genetic sequencing, determining the unique genetic traits, susceptibility, and the resilience to the disease. A new advancement in medical science like predictive diagnosis and the precision medicine. Genomic information is the backbone. This technique has allowed drug makers to come up with the medicine that work on the select group of the individuals based on the similar genetic makeup as against generic drug used to the little success so far. Fine. This is also used for the treatment of the cancer therapy, and in future this will also be used to fix disorders like asthma, diabetes, mellitus, epilepsies, and the Braille phenotype of the autism, attention deficit syndrome. Means the permanent medication. We'll try to find out the permanent medication of what we have mentioned so here. This is the prospect of the genome sequencing. It's the prospect of the genome sequencing. Yes. So the prospect of the genome sequencing in the case of the plant was a drought tolerant, high-yielding varieties, nutritionally enriched varieties. While the prospect of the genome sequencing in the case of the human is more just to try to find out the, try to find out the genome, try to find out the evolutionary pattern, try to find out the mapping, try to find out the cause of disorders, permanent medication of genetic disorder and even the complex disorder. This is the ultimate purpose of the genome sequencing. Fine. So if you just look at the few questions, see here, coming to the question part. See, these questions are based upon the discussion that we have done in the previous lecture. Means in the yesterday lecture, see, genetically engineered brinjal. This was question in 2011. BT brinjal, and the objective is what? It's a pest resistance. So whether it is the BT cotton or the BT brinjal, objective is pest resistant. Yes. Other than resistant to the pest, what are the prospects for the genetically engineering plant has to be created? Enable them to withstand drought, clear. Enable to increase the nutritional value of the produces, fine. To enable them to go and do photosynthesis, spaceship, no, because it's a photosynthesis is the natural ability of the plant. So for this we are not supposed to do anything. To increase their shelf life, to increase durability, fine. So see here 1, 2 and the 4th options are correct option, and that was the question in 2012. Again you see here the recombinant DNA technology, which involves gene to be transferred across the different species of the plant, animal to plant, microorganism to the higher plant. Okay. At present, scientists can determine the alignment or the relative position of the gene. Means it's a genomics. So I'm just telling you, you have already, I have shared all these questions to everyone in PPTs, in PDF form. Clear? So you have already these questions. I'm just showing you so that you feel comfortable, to what extent this topic is important for you. So at present, scientists can determine the alignment or the relative position of the gene or DNA sequences of the. How does this knowledge benefit us? It is possible to know the pedigree of the livestock. Yes, pedigree means generation. If this concept of the genome sequencing could also be used in the case of the plant. It is possible to understand the cause of all human diseases. Yes, possible, and it is possible to develop disease-resistant animal breeds. All possible. Right? Okay. So here, let me take here the first application. The first application is what? Gene therapy. Gene therapy. This first application is the gene therapy. That is fixing of genetic fault. Fixing of genetic fault. Fixing of genetic fault. Fine. So before coming to this, please let me know, everyone. Are you getting my reception properly? Are you getting my reception properly? My audio and the visual receptions are clear to everyone? My audio and visual reception are clear to everyone? Audio and visual receptions are clear? saying yes. Except Alex, no. Meenakshi, yes. Mahak, yes. Shreya, yes. Namrata, yes. Everyone is saying yes. It means if you're getting a stuck video, or if you're getting a reception, if you're not able to get the reception properly, this is probably in two ways. One is gene replacement, and the second is gene editing. Gene replacement and the gene editing. This is also called as genome modification. This is also called as the genome modification. So let me take one by one. Like the first is the gene replacement. See here. This is cell with non-functioning gene. This gene is non-functional. So here we need to replace healthy gene. Clear? And as a result of this, now this cell is going to have healthy gene. So here what happened? Faulty gene is replaced by healthy gene. Faulty gene is replaced by healthy gene. Faulty gene is replaced by healthy gene. Means we have simply changed, we have simply replaced the entire gene pool of the faulty gene by healthy gene. That is the gene replacement. Okay, that is the gene replacement. Right. Fine, that is the gene replacement. Fine. Second is genome editing. Correction in